Hello and welcome to Cambridge Biomedical's Flow Cytometry Techniques video. My name is Dr. Sonal Gupta and I will be taking you through the flow cytometry process we utilize at Cambridge Biomedical. This will be a hands-on demonstration and will be showing our state-of-the-art BD Fax Canto 2 systems in operation. In our previous videos, we have discussed the sample handling process within Cambridge Biomedical and how it is essential for data integrity. This process is common to all of our bioanalytical assay techniques, and hence we will just cover the flow cytometry process in this video. We hope you enjoy our video. Now let's start on our journey.
Hi, my name is Malloy Mangada and I am a research scientist here at Cambridge Biomedical. I have 15 years experience in flow cytometry and more than 20 years experience in a clinical lab. Flow cytometry has several distinct advantages. It has the capability of identifying cell populations producing analytes of interest. It also has the ability to measure analytes on the cell surface or inside the cell. It can also detect up to 18 analytes or parameters simultaneously, and the newer generations can detect up to 30 analytes or parameters. Flow cytometry has also high throughput, up to 100,000 cells per second. One of the distinct advantages of flow cytometry is that it can isolate and identify different populations of cells for further investigation. So there are several issues that you will encounter when working with flow cytometry. The first is that you will need a higher level of expertise. A lot of training is required when learning and understanding flow cytometry. The equipment is expensive and can range from $10,000 for a very basic unit to over $500,000 for advanced systems that can measure over 30 parameters. There has been no established or universal validation procedures until very recently, so there is a lot of homebrew experiments, but none that are validated or qualified by the FDA. Another issue is that it requires relatively larger amount of samples as opposed to other common assays that use serum or plasma in very small quantities. So, to compare flow cytometry to other more common assays in the lab, such as immunofluorescence and ELISA, flow cytometry can measure up to 18 or more analytes. Immunofluorescence can measure up to 3 analytes, while ELISA can be multiplexed, but for the most part you only measure 1 analyte. Again, flow cytometry has a high throughput, can measure many cells per second, while immunofluorescence you are limited by what is on the slide, and it's either a manual analysis using a microscope or using imaging software. ELISA has the advantage that there is a lot of automation widely available. Flow cytometry is a valuable technique and is used in cancer research and clinical trials, in immunology, infectious disease, vaccine research, microbiology, molecular biology, and marine biology. And now I'll hand you off to Dr. Sonal Gupta, who will show you the process in the lab.
This is the start of the sample processing part of the assay, where we split cells in the cell culture biosafety cabinet. Here we pipette suspension cells into 50 ml Falcon tubes for centrifugation. Here we are programming the centrifuge to spin the cells at the required speed and temperature for 5 minutes. We will stain the resulting cell pellets with fax antibodies. Here we prepare the antibody cocktail for staining various cell populations using different combination of fax antibodies. Then we add the appropriate volume of the antibody cocktail to each sample and incubate on ice in the dark. Here we load the sample tube on the SIT or the sample probe for acquisition by the fax Cantor 2 in 5 ml glass fax tubes. The flashing lights indicate that the instrument is acquiring the sample and analyzing it using the fax Diva software. Thank you for watching this video. We hope that it has been informative. We welcome comments and suggestions for other topics. Please email us at marketing@cambridgebiomedical.com. Thank you again.